the study we presented at ASCO is the CHRONOS study, which is a phase 2 study of anti-EGFR rechallenge with panitumumab in metastatic colorectal cancer, driven by liquid biopsy assessing circulating tumor DNA. So the rationale of the study is that we previously described that resistance under therapeutic EGFR blockade with monoclonal antibodies in metastatic colorectal cancer is driven by expansion of RAS and EGFR ectodomain clones. And the good thing is that you can monitor this in the blood through circulating tumor DNA by liquid biopsy. And there is a decline in resistant conferring alleles upon therapy withdrawal. So this pulsatile behavior provides the rationale for a rechallenge when you have the clearance of this tumor mutation that confers resistance in blood, and thus the tumor regains sensitivity. Yes, CHRONOS is a phase 2 study evaluating a patient with performance status 0 to 2 that should have responded to a previous line of anti-EGFR treatment. We monitored, we assessed in blood circulating tumor DNA for mutation of EGFR, ectodomain, RAS, and BRAF by digital droplet PCR at the moment of rechallenge. And only if we found complete clearance of these mutated alleles, the patient were allowed to enter the study and receive panitumumab at standard dose until progression. As for the statistics, the primary endpoint was objective response rate, and 6 responses were required to declare the study positive. First of all, we found at the molecular screening that 30% of patients still presented with mutated RAS, BRAF, or EGFR ectodomain mutated alleles. These patients were excluded from treatment. So we treated eligible patients without this resistant conferring mutation, and out of 52 patients, 27 patients displayed this clearance of mutated clones. Patients were treated with panitumumab, and we observed a 30% response rate in this selected population without resistant conferring mutations, and also an additional 30% of patients displayed a stable disease that was sustained beyond 4 months, a sustained progression disease, accounting overall for a 60% of control of disease. I think that results of CHRONOS are important because it's the first study with an interventional liquid biopsy driving decision-making for treatment in stage 4 disease. And also clinicians do use rechallenge with anti-EGFR monoclonal antibodies in clinical practice. However, this is mainly based on clinical characteristic of patient, that is previous response and an appropriate time interval between previous anti-EGFR administration and rechallenge. Now with CHRONOS, we provide a valuable tool that is liquid biopsy for monitoring ctDNA for resistant conferring mutations that actually is reflecting the real status of the tumor at the very moment of rechallenge. So I think that this approach has got several advantages over the clinical-based rechallenge. First of all is to exclude those around 30% of patients still displaying resistance conferring mutations. Then we observed a response rate that is in the range of 30%, that is higher than previously described, and it also favorably compares with other therapeutic options in this line of treatment. And overall, this observation suggests that liquid biopsy can be fruitfully incorporated in the clinical management of metastatic colorectal cancer.